Hello, everyone, and welcome to this webinar on multidrug resistant gram-negative bacilli. It's my pleasure and honor to chair this program, which is in collaboration between the ISAC, International Society of Antimicrobial Chemotherapy, and the Japanese Society of Chemotherapy. I'm honored to be with you today, and thank you for joining in. We have 6 presentations on a very hot topic of multidrug resistant gram-negative bacilli. As you know, these gram-negative bacilli, multidrug resistant, have been classified by the World Health Organization as critical priority pathogens based on their contributing morbidity, mortality, the complications that they can endure, as well as the low number of potential therapeutic options. And for this, we have 6 speeches today to cover various aspects of how to deal with these difficult pathogens. And without further delays, I'd like to introduce our first speaker, Professor Juliette Severin. Professor Severin is a clinical microbiologist, associate professor, and medical coordinator of the Infection Control and Prevention Unit at the Department of Medical Microbiology and Infectious Diseases at Erasmus Medical Center in Rotterdam. She is currently the vice chair of the National Health Associated Infections and Antimicrobial Resistance Monitoring Group and the chair of the working group that is updating the Dutch infection prevention guidelines on highly resistant gram-negative pathogens. Juliette will speak to us on how do we define multi-drug resistant gram-negative pathogens. Professor Severin, the floor is yours. The microphone is yours.
So good afternoon for those in Asia and good morning for those in Europe. It's a pleasure to speak in this very nice webinar. And I have the difficult task to talk about definitions on MDR gram-negative bacilli. So I will start with my disclosure, first one on my research collaboration with bioMérieux, but more importantly, my focus is on infection prevention and control. And I only have 10 minutes, and I will definitely not end up with a nice list of definitions. I will start with this landmark paper that was published in 2012. And actually one of the next speakers is a co-author on this paper. And this was an expert group that was proposing definitions for interim standard definitions for acquired resistance, for multi-drug resistant, extensively drug resistant, and pan-drug resistant bacteria. So quoting the paper, in the strictest sense, multi-drug resistant microorganisms are labeled as multi-drug resistant because of their in vitro resistance to more than one antimicrobial agent. And of course, if you stick to this very strict definition, a lot of bacteria will be classified as MDR. Well, the importance of this paper, I think, was obvious. There were many definitions circulating around, and this was an attempt or an important step to improve the comparability of surveillance data for these organisms and to better assess their global, regional, and local epidemiological importance and public health impact. I underlined the word interim. I don't think that there is an update after this published, but I think it's still, well, as I said, a landmark paper. The definitions that were finally published were these. for the gram-negatives. So the isolate is non-susceptible to at least one agent in 3 or more antimicrobial category listed in a separate table. That's for MDR. For XDR, the isolate is non-susceptible to at least one agent in all but 2 or fewer antimicrobial categories. And pan-drug resistant, as the word says, non-susceptibility to all agents in all antimicrobial categories for each bacterium. So 3 things are important to mention here is, first of all, it's not resistant, that was in the definitions, but non-susceptible, including resistant, intermediate, or non-susceptible when using the clinical breakpoints as interpretive criteria. And as we know by now, the word intermediate has been under debate, has changed in meaning. So this also has an effect on the definitions on MDR and XDR and PDR. The second important comment is that each antimicrobial category in this definition is equally relevant. And of course, it's the question if that is, that's still holding true at this moment, still correct. And the third is that bacterial isolate should be tested against all or nearly all of the antimicrobial agents within the antimicrobial categories. And we know that laboratories are tending to work cost effectively. Also, commercial panels do not include all agents and all categories. So that is difficult to fulfill, to use these criteria. Is it important, these definitions? Well, if you want to use it as surveillance, public health, yes, it's important, but not everybody is using these definitions from 2012. And we know that a lot of countries have made their own definitions, and these are usually mainly to guide IPC measures. And what the effect of this was nicely shown in this paper from the group of Zurich. And I'm showing you one picture of this, one figure. They looked at Klebsiella pneumonia isolates from a hospital, and they compared the definitions. First of all, the ECDC MDR definitions from the paper I just showed you. These were for public health surveillance purposes made. And then the CRINCO and the CRINCO definitions are national definitions from Germany used to guide IPC measures, and the University of Hospital Zurich, so a hospital definition, set of definitions also to guide IPC measures. And what you see nicely in this figure is that, of course, you will get different percentages of MDROs. And even when the purpose is to guide IPC measures, then you will see that there is a difference. Then there is another international important paper that was already mentioned by our chair of today. It's from the WHO. It's currently been updated, was published in 2017. And the purpose of this list, and it was the priority list of antibiotic-resistant bacteria, was to guide research, discovery, and development of new antibiotics. And they used a very nice set of criteria, 7 in total. I listed them here. And these guided these priority lists. And this was the result. And as you see, the critical are all gram-negatives. And then it's the outcome was more a drug combination that was defined as important. So this was used for R&D investment mainly. That was also the purpose of this priority list. But I think most of you will recognize that it also emerged as a tool for raising AMR awareness, informing IPC interventions, and interventions for antimicrobial stewardship. So this bug-drug combination approach is nice and was also used in this paper, recently published in The Lancet, the Graham paper on the global burden of antimicrobial resistance. And they looked at 23 pathogens and 88 pathogen drug combinations in 204 countries. So avoiding these more difficult definitions of MDR, XDR, and PDR. And I listed here the pathogen drug combination with most deaths. And these were 7 in total. MRSA was on top, but in this top 6 list, there were again 5 bug drug combinations for gram negatives. I was actually surprised that the fluoroquinolone resistant E. coli also appeared here. A short word on the Dutch definitions on highly resistant microorganisms. So we have these definitions and we only use them for IPC measures. And what I like is that it's partly drug-bug combinations, but also some combinations of antibiotic classes and resistance to these classes of antibiotics. So all Enterobacterales that are ESBL positive are defined as highly resistant microorganisms. Also all Enterobacterales that are either carbapenemase positive or carbapenem resistant. And as a special HRMO definition in the Netherlands, and I think there is not a single other country that's using this, is the combination of resistant against one of the aminoglycosides and the quinolones. So if an E. coli is resistant to gentamicin plus ciprofloxacin, it is defined as a highly resistant microorganism, and we take measures. Another antibiotic, bug drug combination that we have in the Netherlands is the Stenotrophomonas multifilia that is resistant to cotrimoxazole. This was published and defined already in 2005 for hospitals and in long-term care facilities in 2019. And we still have the same definitions, but these are currently being updated. So the former definitions were based on these 3 criteria. So the bacterium has to cause disease, has an acquired resistance, possibly in combination with intrinsic resistance, which hinders first choice therapy, and as a third, bacteria can spread if, besides basic hygienic measures, no additional IPC measures are taken, or where it has been shown that in daily practice, despite basic hygiene measures, spread occurs. And now in the revision that we are now doing, we have added that it can be a plasmid even that we have to worry about that can spread. And why we think that's important, I just as a teaser, but I'm sure that the other speakers will probably also raise this difficult resistant mechanism. So this is an E. coli and a result from the Phytec-2. And as you can see, sorry, it's in Dutch, the antibiotics, but you can understand it's all sensitive, all very low MICs. And the question that I'm raising here is this a multidrug resistant gram-negative bacteria? Well, just basing on the definitions, for example, by the WHO or by the paper from 2012, it is not. But I'm not revealing here the ertapenem MIC, which was elevated, and this was an OXA48-like, a positive E. coli, and we know that this is on a plasmid. So in the Dutch guidelines, we say this is an MDRO, although it's completely sensitive in our AST panels, but this is something that we do not want to have spread in our hospitals. And it's important that we have these definitions at least in the local situation, because as we know is that patients travel around and in hospital networks, nicely pictured on the right for the situation in England, and on the left is the situation in the Netherlands. Academic hospitals are really hotspots where patients gather, and also multidrug resistant pathogens gather. And when patients are transferred from one hospital to another, we have to be sure that we speak the same language and we take the same measures. Otherwise, we will spread our pathogens, our multidrug resistant pathogens, but also patients will get very confused. So to conclude, regarding definitions for MDROs or multidrug resistance, gram negatives, the purpose or the goal matters. I showed you it can be for public health purposes, R&D purposes, IPC purposes, but also maybe for treatment purposes and clinical trials. setting matters. I did not mention the One Health concept, but these definitions are really focused on human health, and we have to think also about the animal and environmental domain, and the lab capacity matters, as I explained before. So bug-drug combinations, resistant mechanisms, and plasmid presence, yes or no, is maybe more value than the definitions for MDR, XCR, and PDR, but that is probably open for a discussion.
Thank you very much, Dr. Severin. And we can now move on to the next presentation by Professor David Paterson. I'd like to remind the participants that we will address all your questions at the end of the webinar. And you can submit your questions by typing them on the right side of your screen, and we'll try to answer as many as possible. It's now my pleasure to introduce Professor David Paterson. Professor Paterson is the director at the University of Queensland Centre for Clinical Research. He's also a consultant infectious diseases physician at the Royal Brisbane and Women's Hospital. He is Australia's most cited infectious diseases physician and is the country's highest cited researcher in the field of microbiology. His research focuses on the molecular and clinical epidemiology of infections with antibiotic-resistant organisms, with the intent of translation of knowledge into optimal prevention and treatment of these infections. Professor Paterson will speak to us on mechanisms of resistance. David, the microphone is yours.
Thank you very much for that lovely introduction. And I have to say, it's a great honor to speak in front of so many people, even though we're not all face-to-face. And in fact, just in the last weeks, I've just moved to the National University of Singapore just to really get some interesting perspectives on multi-drug resistance. So as Souha said, I'm from Australia, and I think Australia is the only country in the world where the discovery of antibiotics is celebrated on a dollar bill. This is our old $50 bill, and you can see Fleming's original agar plate. This is Florey, who developed penicillin, but then you can also see the agar plate, which did actually demonstrate there was non-susceptibility to at least one of the discs there that you can see in the middle of the screen. Now, I'm a clinician, and I'd like to just present a case to you, just to sort of put some of these aspects of mechanisms of resistance into perspective. So this is a 65-year-old man who's actually got septic shock. So his past medical history there you can see, including type 2 diabetes, hypertension, a smoker, and he comes to hospital with chest pain and then soon after has a cardiac arrest. In his second day of ICU, he is defined as having pneumonia, and they're actually calling it hospital-acquired pneumonia and giving the patient meropenem. Now, I'm not sure all of us would agree with that treatment choice, but that's what happened. He did go on to have coronary artery bypass grafting, but because of his smoking, and he probably did have some unsuspected chronic airways disease, he had a very slow wean from the ventilator. And in fact, on the 14th day, while still in ICU and mechanically ventilated, he has a fever. There's an increasing amount of green sputum. There is deteriorating oxygen saturations. You can see he's sick. He's got a high fever and a low blood pressure. And there is radiologic evidence of pneumonia. And I'd like just to pose to you, just for you to think about, well, in this setting, this patient has now been in hospital for more than 2 weeks. What would you use as treatment before you get the answers back from the microbiology laboratory about that green sputum? And these are some of our options. Now, it's not as pretty a susceptibility profile as what we saw in the Netherlands. In fact, this Klebsiella pneumoniae, which grew from the endotracheal aspirate, was resistant to all of these antibiotics you see, and that includes meropenem, it includes ceftazidime avibactam, as well as the others I've listed there. And so I guess the key element in my talk today is what might be causing this resistance? What is the mechanism of antibiotic resistance in this particular scenario? And I'd like to draw your attention to a paper that only came out 2 weeks ago. It's online. I don't believe it's in a print version yet. And it talks about some consensus on beta-lactamase nomenclature, because those of you who are working in microbiology or infectious diseases know for sure that if you see an organism with a susceptibility profile like this, there is going to be some sort of nasty beta-lactamase. But many of us get very confused with beta-lactamases. And why is it such a confusing field? Well, in fact, the trouble is microbiologists were left to their own devices. And so beta-lactamases they discovered could be named after the bacteria that it was found in, their particular strain or the plasmid, even the patient. And the classic example there is Mrs. Temonira in Athens in Greece, who had the TEM beta-lactamase named after her. What is the substrate hydrolyzed? What is the geographic location? So, for example, VIM comes from Verona, NDM comes from New Delhi. Now, whether those geographic depictions are right or wrong is another big question. And the trouble was, again, why it's so confusing, no consistency. In fact, there were examples of different names for exactly the same beta-lactamase, and I can give you some examples there. And since we're talking about invited by the Japanese Society there, I'd like to point out Toho University in Tokyo and Professor Ishii and others who discovered that beta-lactamase. Others called it CTXM, but it really just is an example of why we need consistency. And the other really tricky thing is the number of different beta-lactamases, the unique types of them you can see is over 2,000. You know, it'd be great to be studying this field in the 1980s. You'd only have 10 or 12 to think about, but now it's so confusing because there's so many different beta-lactamases. So there are some important rules that have been put forward. There is going to be a sort of a one-stop shop for new beta-lactamase numbers, and they're not going to be sort of based on silent mutations. There's got to be an amino acid change, and they've got to be beta-lactamases from natural sources, not made up in the laboratory. And they're not to be named after geography. There's a naming convention. You can see, for example, the gene that encodes TEM1, the way that is written. And, you know, I think it is important that these rules are going to be in place, and that's going to help all of us talk the same language. Well, let's get back to this particular patient and their Klebsiella. And, you know, what really interests me here is that it's resistant to meropenem, resistant to ceftazidime avibactam. And the resistance to meropenem really implies that most likely there is a carbapenemase. And there are a number of different classes. The class A carbapenemases are what we call the serine beta-lactamases, and that includes, for example, KPC, and also class D, and we've heard from Juliette, OXA48, that quite silent silent killer, that silent beta-lactamase. And then there are also metallo-beta-lactamases, for example, NDM or VIM, which I've already mentioned, and IMP. And one of the reasons, again, why I want to mention IMP is that in fact was the first carbapenemase detected, 1988, in Japan. And that's really how far back carbapenemases go. KPC was discovered in 1996 and reported in 2000, NDM 2006 and reported in 2009. But clearly these are a massive issue for us today. And one of the really important things for us is to, make these mechanisms, how do we make this sort of practical? And in fact, can we predict the antibiotic resistance mechanism from the susceptibility profile? And so for me, if I see Enterobacterales resistant to meropenem, that implies that there is the presence of a carbapenemase. And if there is resistance to ceftazidime avibactam, that implies that there is a metallo-beta-lactamase, an MBL, present. Now, these are not 100% rules, but this is really, if I was walking in a clinical microbiology laboratory and I was teaching one of my colleagues, this is really important signals to me as to what the mechanism of resistance is. So because we've seen that there is resistance not just to meropenem, but also ceftazidime avibactam, that implies to me that there is a metallo-beta-lactamase. And with any enzyme, we talk about the substrate and also what the enzyme might be inhibited by. And these metallo-beta-lactamases destroy penicillins, cephalosporins, and carbapenems. But interestingly, they do not destroy aztreonam. They are inhibited by EDTA. Now, we can't really put that into clinical use in patients, but interestingly, they are not inhibited by clavulanic acid, and importantly, they're not inhibited by the new beta-lactamase inhibitors like avibactam or relibactam or vaborbactam. And therefore, it's not surprising that anything that will be producing a metallo-beta-lactamase will also be resistant to ceftazidime avibactam. So I mentioned before some of the examples of IMP and VIM. Those of you in Brazil would be familiar with SPM, NDM, and there certainly is a worldwide distribution, but they're particularly common in the Indian subcontinent. But there are some really sort of unusual geographic sort of call out. So for example, in Australia, we have a lot of IMP4, and in various other countries, there are some geographic specialties. So our treatment options for metallo-beta-lactamase producers are things like polymyxins, cefiderocol, and there is also a combination which you can make yourself, which is ceftazidime avibactam with aztreonam. And the reason why aztreonam is in there is that the avibactam is destroying any other beta-lactamases, but the aztreonam will not be destroyed by metalloenzymes. They're some of the treatment options, but even here we have potential mechanisms of resistance. Now, I won't go into details about, for example, colistin resistance, but you can see that it really disrupts the membranes, particularly the outer membrane, but it can also disrupt the inner phospholipid membrane. But mechanisms of resistance mean that it cannot disrupt those membranes. And we also have, I think, of less clinical importance, the plasmid-mediated mechanism, MCR. Now, cefiderocol is what we call a siderophore cephalosporin. So we know that siderophores are so important in bringing iron in through the outer membrane of gram-negative bacteria. And what cefiderocol does is it sneaks in through those ion channels through the outer membrane. It can get then very efficiently, without being destroyed by beta-lactamases, and bind to the penicillin binding protein. So that's really how cefiderocol works. So I always sort of think of it as like the Trojan horse. It's sneaking in, the body thinks it's bringing in iron, but in fact, it's bringing in this cephalosporin that's going to do bad things. So there is an interesting new mechanism of resistance to both cefiderocol and also to aztreonam with avibactam. Now, even though that's not available clinically, it's interesting that the mechanism of resistance has been determined even before it's out there, and that is that in E. coli, there can be an unusual insertion sequence in PBP3. So normally, we think of PBP changes, penicillin binding protein changes, with MRSA, for example. But here is an important gram-negative resistance mechanism in E. coli, where this insertion sequence now leads to an inability of either cefiderocol or aztreonam to bind to the penicillin binding protein, and therefore we may actually have resistance in E. coli. Now, sometimes, as a result of other beta-lactamases, we can also get compromise of these antibiotics. And of course, the million-dollar question is, could the bacteria get clever and switch off the ability of cefiderocol to sneak in through those ion channels through the outer membrane? So there's a lot to be learned about these new antibiotics and their mechanisms of resistance. So if we were to put all of this together, mechanisms of resistance in gram-negatives, beta-lactamases are far and away the most important. We now have emergence of these PBP changes. We also have long had loss of outer membrane proteins. And I think of these as sort of the doors through which many antibiotics get through the outer membrane. And I guess very classically with Pseudomonas and imipenem, that door can be closed. We can have loss of those outer membrane proteins, and that can be a mechanism of resistance. This often goes hand in hand with efflux pump overexpression. So in this way, we have active pumping of antibiotics out of the bacterial cell, contributing to often multi-drug resistance. Now, just say we had this Klebsiella, it was meropenem resistant, but it was not resistant to ceftazidime avibactam. Then we're likely to have either a KPC type of carbapenemase, so in other words, one which is inhibited by avibactam, so KPC or OXA48. And I do want to point out, as Juliette has told us, that sometimes we may not actually get that trigger of resistance and yet still have OXA48 and some of its derivatives present. So that's an important conundrum for us. What is the significance of the gene without the phenotypic mechanism, the phenotypic expression of resistance? So KPC producers very widespread in the US, also in places like Colombia, Italy, Greece, and I have to say China as well. Whereas the NDM producers, as I mentioned earlier, through the Indian subcontinent, but many, many other parts of the world have been encountering NDM producers. And similarly, OXA48, Indian subcontinent, Mediterranean, North Africa, Middle East, Europe, but again, we're finding those all over the world. So the geographic boundaries have certainly been broken down. Now, I'd like to finish just by briefly mentioning mechanisms of resistance in something that you might think is a little bit simpler. You know, we've only got resistance to ceftriaxone and also gentamicin, ciprofloxacin, and trimethoprim sulfamethoxazole. So we don't have resistance to carbapenems. We have susceptibility to ceftazidime avibactam. And of course, in this situation, we are very likely to have an ESBL, an extended spectrum beta-lactamase. They've been around more than 40 years. They're typically in the Enterobacterales. And when we say an extended spectrum, that originally meant compared to the parent TEM1 or SHV1 enzymes that will just knock out ampicillin. So their spectrum of resistance was broader than the parent beta-lactamase and now included third generation cephalosporins. Of course, now the most frequently found ESBL worldwide is the CTXM type ESBL. And we always need to remember that on the plasmids, there may be the genes encoding the ESBL, but often resistance to other antibiotics. Those mechanisms, those genes are encoded on the same plasmid. So unfortunately, antibiotic resistance is a complicated business. And I know many people, including infectious diseases specialists, sometimes go a bit pale when people start talking about TEM 181 and NDM 16. But I think it is important that we do recognize the major types of beta-lactamases, their inhibitor and substrate profile, because that does determine the resistance phenotype. We also have to remember that typically strains now have multiple beta-lactamases in the one type. And one thing we found very relevant in the Merino trial was that even though we were seeing ESBLs, and you would think, for example, that piperacillin tazobactam should be active against many ESBL producers, often these ESBL producing strains also produced OXA1, which is actually not inhibited by tazobactam and led to a compromise in piperacillin tazobactam's activity. The world is complicated. We do have other resistance mechanisms, the outer membrane protein loss, the efflux pumps, and now the PBP changes. So an interesting world, but thank you very much.
Thank you very much, Professor Paterson, for simplifying a very difficult topic. And again, we're gonna leave the questions till the end of the webinar. So it's now my pleasure to introduce our first colleague from Japan, Professor Katsunori Yanagihara. He's a professor at the Department of Laboratory Medicine at the Nagasaki University School of Medicine in Japan. Since 2017, he has been research leader at the Japan Agency for Medical Research and Development on diagnostic methods and therapies to contribute to measures against antimicrobial resistant bacteria. So Professor Yanagihara is going to talk to us about rapid molecular diagnostics for gram-negative pathogens.
Thank you for introducing me, Professor Kanji. I'm very happy to speak in an important webinar. So just story, before 1914, infectious disease was first cause of mortality. Now post-antibiotic era, I'm showing that MRSA, ESBL, TB, and there are important pathogens as resistant pathogens. Also, in introduction, Dr. Kanji also mentioned that WHO concerning that the antimicrobial resistance is a global report on surveillance. So what did I do to protect antibiotic resistant in Japan. We started National Action Plan 2016 and it finished 2020. It pointed the pandemic awareness and education and surveillance monitoring and infection prevention and control and appropriate use of antimicrobials and research and development, international cooperation. So this is the results. So after 2020, so their group checked that the changes in antibiotic sales in Japan. As you can see, this is the 2016. The sales is decreasing. But 2020, it's a coronavirus pandemic. So most patients didn't go to the hospital. It is some reason why the antibiotic sales is decreasing. So we have to follow in the future. And this is also that national data. You can see that 60.7 million samples and 5.9 million patients. Also that the data from 1,300 hospitals. It should be national data. This is the annual trend. As you can see that this red color and pink color is at the MRSA and PSP gram positives. This is at the blue and green gram negative, also yellow gram negatives. So you can see that the MRSA is decreasing, also PRSP decreasing, but gram negative, resistant gram negatives is increasing stable. And this is that we can check, they can check that sample. This is respiratory and urine and blood and stool. You can see that this is the third generation cephalosporin resistant to Klebsiella pneumo. It's increasing. So that means that the gram positive is decreasing, but the gram negative are increasing. I think that it depends on the country. But in Japan, so gram negatives will get more important. So, and the resistant problem, so there are some problems, but I'm focused on overuse and misuse of antibiotics in humans and animals. So why do we need rapid molecular diagnostic? So avoid to use overuse, misuse antibiotics. If you see the patient, the condition is very poor or critical patient, so we have to use the strong and broad spectrum antibiotics. But sometimes it's overuse and misuse. And if you wait the susceptibility after culture data, but we have to use the antibiotics immediately. So it's a serious case, serious patient. So we have to use the broad and strong antibiotics. So if we use the rapid molecular diagnostic, so we can choose the appropriate antibiotics immediately. So this is the data. Usually the step sampling, this is a current method, and ID and AST. You can see that the gram staining, sample preparation, and about 36 to 72 hours. But if you use the rapid ID, NAAT, so 30 to 40 hours. Also in this session, rapid molecular diagnostic, if you do rapid NAAT from sample directly, that will get results at 0.5 to 8 hours. It's much faster than current method. So now you know that the development in laboratory machine for detection resistant pathogens. Also, to perform coronavirus PCR, many hospitals purchased PCR machine. So in with or post-corona era, so we have to use this machine for to detect the resistant pathogen. This is a biomarker and BD and something like that. This is a Japanese machine. So the role of rapid molecular diagnosis for BSI. So usually blood culture positive and sample plate for subculture and resistant testing 48 hours. But if you use it, this is a for the Beirut, if you use it, you get in 15 hours. The Beirut system, you know, this blood culture gram negative and blood culture gram positive. They have two options, to negatives and positives. Beirut system, just review the mechanism. They didn't use that PCR. This is a micro array system. This is target gene gram positives, the Staphylococcus and Enterococcus fecalis and Faecium and Septopneumo. This is gram negative, Acinetobacter, Enterobacter, protein, something like that. There are important pathogen in blood stream infection. How about that identification the organism from clinical blood cultures? This is the E. coli, Klebsiella, pneumo. So we compared that this identification from data, Beirut, and clinical blood culture data. And you can see almost 100% is correct to culture data. How about the ESBL? Dr. David Paterson mentioned that ESBL is one of the most important pathogens in gram-negative bacilli. So Beirut allows us to detect ESBL correctly. So ESB result totally agreed with CTXM result of Beirut. So that means the Beirut is useful for the rapid detection of ESBL. So, and Beirut shortened the turnaround time. This is the bottom column, blue column is mentioned that the conventional method. So as I mentioned previously, we'll get data at day 3. But if you use that molecular technology, you will get at day 1. So use appropriate antibiotics much faster than a conventional method. How about that intervention study with Beirut for rapid diagnosis of BSI? This is a control period and the intervention period. So we compare that the mortality. So in control period, 12.1 to 12.9% mortality, but intervention. So if the using Beirut system, mortality is get lower than control period. So it is a role of molecular diagnosis system for BSI. And in now or future, important thing is how to compare the novel methodology. It's very important because so if you purchase the machine, you have to use several years. You can change that antibiotics immediately, but you cannot change the machine. So before purchase machine, you have to compare that. So the paper mentions that the desirability of outcome ranking management and therapy, that I will explain that. They classify that the antibiotics that this 6 narrow, intermediate, intermediate 2, broad, last resort. This is at the resistant pattern. If all antibiotics are sensitive, so you have to use narrow antibiotics. But when this is a RSSS, you can use the intermediate. So they can, if you use that this pathogen, if you use an intermediate one, so to slight over treatment, and they can culture and they can compare to the method. So that this method is good tool to compare the novel methodology. And this is the data, and they can compare the retrospective proof in concept study. That means that they do that the culture data, and also they use that molecular diagnosis data, and they can compare to the antibiotics. And the data mentioned that the BioFire BCD2 panel is better than BCID and Berizine system. That you can see, that means that if you use a BioFire BCD2, you can use that ideal treatment to the patient. So why this phenomenon happened? That means that the BCD2 panel, that star means that indicate a new target on BioFire 1 panel. That's the additional point. That means if we get more data, so that means that also that bacteria identification, also AMR gene. So we will use appropriate antibiotics immediately to the patient. So we have to investigate more. That is one thought. And how about that pneumonia? This is from that United States investigators. Pathogen detected among others community-acquired pneumonia requiring hospitalization. So this is also that the viruses, also bacteria. So many bacteria is isolated. So as you know, also respiratory infection, especially pneumonia, is very critical. So we have to use antibiotics immediately. This is a BioFire pneumonia panel menu. This is gram negatives. So as you can see, each panel covered most of pathogens in pneumonia. So I think it's very useful. Also that the AMR gene, this is for MRSA. This is also that gram-negative bacilli, also ESBL. So we had better use for that serious pneumonia. And this is also very simple, 2 minutes and 1 hour. You get the data in 1 hour. This is the representative data from that pneumonia panel. They use 200 samples, and the data is BioFire 44.5% bacteria on the viruses of the panel. So, and the main point is standard of care is you have to wait 44 hours and 14 hours. So you have to wait 60 hours to get the results. But if you use a molecular diagnostic, rapid molecular diagnosis, you will get 2.3 hours. So you will start much faster than a standard of care. This is our data. Just explain that this is 15.7 cases. This is pseudo and RS, Klebsiella pneumo. This blue bar mentioned that culture and BioFire means a rapid diagnosis system, molecular diagnosis. This is both. And this is a BioFire panel only. That means that sometimes we cannot culture because the amount of bacteria is not enough. We cannot culture. But the molecular detection system, its sensitivity is much better. So this data indicated the this biofilm panel data is reasonable because that it's covered all cultured bacteria. So it's one data. And Maruji also, this is also molecular diagnostic and rapid detection of resistant pathogens. So as you know, this is a Maruji top workflow. This is a culture plate put on the circle and the laser irradiate to the flow. And this is a spectral pattern. This is Staph aureus, E. coli, and Stenotrophomonas. So how do you use that detect to the resistant pathogens? So this is culture plate. We use the supernatant and antibiotics is mixed cultures. So if the supernatant contains that beta lactamase changes structure of antibiotics. So that means that this is a data. This is control. The supernatant contains the beta lactamase. This peak is changed, so we can detect. So this is a data. This is a non-producing K pneumoniae, and this producing K pneumoniae. This hydrolyzed means that it's contained that the ESBL and ESBL change to the structure of the antibiotics. This sensitivity and specificity is better. Also, that this is meropenem to carbapenemase, so the similar data. It's mentioned that the allergy is useful, may be useful for the detection of resistant pathogens. Which directions with the this is a current method, and if you use that after day 2, that you get 3 hours. And in the future, this is that other group published in Nature Medicine. They use that also Maruji and also artificial intelligence, so machine learning. So they collected that huge data, and that they can analyze that huge data, and they conclude this data. This is that it's mentioned that if you use that Maruji, and if you data a lot of data, and AI study the data, it gives us correct data to use to get resistant pathogens. Okay, I talk about the rapid molecular diagnosis for resistant pathogens. So in now and the future, for appropriate treatment, rapid molecular diagnosis very useful. So we have to use correctly. Thank you.
Thank you very much, Professor Yanagihara, for addressing the important topic of rapid molecular diagnostics. It's now my turn to speak, so I'm gonna share my screen. Okay, and so again, my name is Souha Kanj. I did not introduce myself at the beginning because I thought I'll do that when I speak. I'm a professor of medicine and head of the Division of Infectious Diseases at the American University of Beirut Medical Center. I'm also on the board of ISAC, and it's my pleasure to talk to you on treatment of MDR gram-negative pathogens and introducing the new drugs. These are my disclosures. And so what I'll try to address in this presentation is carbapenem-resistant Enterobacterialis, which I'll refer to as CRE, carbapenem-resistant Pseudomonas aeruginosa, CRPA, and carbapenem-resistant Acinetobacter baumannii. And as you know, all these have been classified as critical priority pathogens. Now, whenever we want to talk about antimicrobial stewardship, we need to consider that effective treatment should be administered as soon as possible. And therefore, we've heard about the increased need for rapid diagnostics. And so part of the stewardship is to consider that the novel antibiotics expand our armamentarium for managing these multi-drug resistant gram-negative pathogens. And we should not be leaving them on the shelves only to bring them when a patient failed therapy. The novel drugs have showed clinical efficacy to comparators in randomized clinical trials, as well as evidence from real-world data. And in some cases, they showed better results in subgroup analysis and much better safety profile. So let's talk about predictors of hospital mortality in gram-negative bacteremia. This is a study that was done and published in 2014. And as you see in red, the predictors are listed, but the most important predictor of mortality is receiving inappropriate antibiotic therapy. And in the same study, they found that having a multi-drug resistant pathogen had the highest odds ratio of 13.05 to correlate with inappropriate antibiotic therapy. So when a patient turns out to have a multi-drug resistant gram-negative pathogen in bacteremia and septic shock, it is likely that they will end up receiving inappropriate therapy. So in addition to the old drugs that we've had in our armamentarium, now we have several novel drugs. Some of them are beta-lactams, such as the combinations of beta-lactams, beta-lactamase inhibitors, the cefiderocol that you've heard about in the previous presentation, as well as cyclin. And as you see from this color-coded slide, you have the spectrum of activity of each one of them. So for example, ceftolozane tazobactam, mostly for ESBL and Pseudomonas, whereas the next three on the list, they have expanded activity, including against carbapenem-resistant Enterobacterialis. And for each one of these new drugs, we have advantages. Ceftolozane tazobactam, as mentioned, is a very good drug for multi-drug resistant Pseudomonas. Ceftazidime avibactam has been approved for empirical as well as targeted therapy. Meropenem vaborbactam seems to create less resistance than ceftazidime avibactam. Cefiderocol has activity against Acinetobacter baumannii. And eravacycline, for example, is a more broad spectrum with activity against gram-negative, but as well as gram-positive. So let me start by addressing carbapenem-resistant Enterobacterialis. And you've seen already the epidemiology of how this organism has spread throughout the world. And if we look at the mortality of treatment of CRE infections, the older studies on the top part of this table showed the very high mortality, in some cases exceeding 50%. Whereas the more recent studies that have used the novel antibiotics have shown much lower mortality, as you see, 8%, 15%, and 9.5%. And with the older drugs, a lot of the recommendations suggested that we really need to use them in combination, whereas we're going to see that for the novel antibiotics, we might not need combination therapy. I put on this slide the clinical studies that have been done to address the ventilator-associated pneumonia and hospital-associated pneumonia. using the antibiotics that potentially have carbapenem-resistant Enterobacterialis coverage. And you see here, you have ceftazidime, avibactam compared to meropenem. You have cefiderocol compared to meropenem, imipenem, relebactam versus piperacillin tazobactam. And in every one of the studies, the new novel antibiotic really showed non-inferiority to the comparator. But what about pathogen-directed clinical trials? The novel antibiotics, we have on this slide 2 of them. The first one compared cefiderocol versus best available therapy against carbapenem-resistant gram-negative infections, and that included carbapenem-resistant Pseudomonas, as well as Enterobacterialis. And in the RESTORE-IMI trial, it was comparing imipenem, cilastatin, relebactam versus a combination of imipenem, colistin against imipenem-resistant pathogens. And again, as you see, the results were comparable to the compared agents, which was best available therapy. And I'm going to detail some of these. Now, another couple of studies that have been done as pathogen-directed clinical trial. The first one was the REPRISE trial, ceftazidime-resistant gram-negative pathogens, where ceftazidime avibactam was compared to the best available therapy. And again, as you see here, the results. And I will detail more the TANGO-2 trial, which was meropenem-vaborbactam versus best available therapy. And why did I include to detail it in this presentation? Because that's the only drug on the tables I've showed you that was particularly directed against only CRE infections and not including other pathogens such as Pseudomonas or others. So let me show some of the real-world data first with ceftazidime avibactam. This is data from Pittsburgh that looked at ceftazidime avibactam, as well as various combinations, including carbapenems with aminoglycosides or carbapenem with colistin, and these are the numbers here. And what they looked at is clinical success defined at 30 days as survival, resolution of signs and symptoms, sterilization of blood cultures within 7 days, and absence of recurrent infections. And if you see in the arm that received ceftazidime avibactam, 85% success, whereas for the other 3 arms, it was at most 48%. And when they looked at multivariate analysis at factor associated with clinical success at 30 days, receiving ceftazidime avibactam had a p-value of 0.03. Ceftazidime avibactam is also used as salvage therapy for OXA48. You've already heard about this mechanism of resistance from the earlier presentation. And again, in this small cohort of patients, 57 patients, the clinical cure and the microbiological cure were 80% and 67% respectively. But when we treat a patient with carbapenem-resistant Enterobacterialis, not only we need to look at efficacy, but also at toxicity. And this is data from David Van Duyn from North Carolina. And as you see, when he compared patients who received ceftazidime avibactam versus colistin, the patients who received ceftazidime avibactam were less likely to die, more likely to be discharged from the hospital within 30 days, but very importantly, they were more likely to experience an overall better outcome, including less renal failure. Now, do we need to use a combination therapy for ceftazidime avibactam versus the monotherapy? This is really a summary of multiple studies that have been shown. And as you see in the light blue, monotherapy had very similar results to combination therapy. So unlike the older drugs where we had to use combination therapy with the novel antibiotics, perhaps combination therapy is not necessary. Now, one thing we need to keep in mind is that in real world study, we need to see what type of problems we could be encountering with using various drugs. This is from Mario Tamburello. He gathered data on a large number of patients, 577, with KPC, Klebsiella pneumonia. As you see, a large number had bloodstream infections as well as respiratory infections. What we learned from the study is that the 30-day all-cause mortality was 25%, and that dose adjustment of renal impairment was associated with worse outcomes. The severity of illness and lower respiratory tract infections were associated with higher rates of death. And really, the combination therapy did not improve mortality versus monotherapy. And there was a large number of patients who received combination therapy. But one thing we learned is that extended infusion did improve mortality, as we see in this curve. And that's why it's recommended to now give it over 3 hours. But 4% of the patients developed resistance. So this is something that we need to keep in mind in patients who receive ceftazidime avibactam. Now, the other options that we have for treatment of KPC, I'm including here meropenem vaborbactam. Vaborbactam is a boronic acid beta-lactamase inhibitors and inhibit other class A, so KPC, CTXM, SHV, and TEM. It's FDA approved for complicated UTI. It was approved in 2017, and it was EMEA approved in 2018 for the complicated UTI, for pneumonias, as well as secondary bacteremia, as well as for treatment of infections caused by gram-negative organisms with limited options. Let me go a little bit over the details of the TANGO-2 trial, and this trial looked at efficacy and safety of meropenem vaborbactam to best available therapy. And again, it was targeted only to patients with carbapenem-resistant Enterobacterales. It was a multi-center study, phase 3, 27 hospitals in 8 countries participated. And I'd like to point your attention to 2 things on these tables, the infection types, and as you see, a large number of patients had bloodstream infections, but the best available therapy regimens that were compared to meropenem vaborbactam, as you see, some patients received monotherapy, dual therapy, even some patients received 4 or more drugs with the combinations that we are used to use in practice, carbapenem, aminoglycoside, polymyxin, tigecycline, and so on and so forth. And these were the results of the TANGO-2 trial. Meropenem vaborbactam showed higher clinical cure rates at the end of therapy and the test of cure. Meropenem vaborbactam is in the light blue, and the best available therapy is in the darker blue. When they looked at day 28, all-cause mortality in the microbiological carbapenem-resistant Enterobacterales modified intent to treat, as you see, the mortality with the meropenem vaborbactam group was lower, not statistically significant, but numerically lower. And I think it's important to know that the study was stopped earlier because of these very encouraging results for meropenem vaborbactam. So if we compare ceftazidime avibactam to meropenem vaborbactam from the Pittsburgh experience, from the North Carolina, Florida experience, as you see, similar results, slightly better result with meropenem vaborbactam. But as I mentioned before, with ceftazidime avibactam, we need to keep in mind that the rate of resistance development is higher than in patients who receive meropenem vaborbactam. You've heard about the metallobeta-lactamase producing Enterobacterales. These are very challenging. And you've heard from Professor Paterson that currently what we've been using is ceftazidime avibactam plus estrionam. And in this study that looked at bloodstream infections with metallobeta-lactamases, you see that in patients who received the combination, the drug-induced acute kidney injury was low as opposed to the other active antibiotic, OAA. The 30-day mortality was much lower, 19 versus 44%. And clinical failure at day 14 was also lower, 25 versus 52%. These were the other antibiotics that were used. And if we look at this curve here, the cumulative survival was much higher for ceftazidime avibactam plus aztreonam with a significant p-value and by Cox regression analysis of factor independently associated with 30-day mortality. Having received ceftazidime avibactam with aztreonam was very favorable. This is a very nice review that was just published a couple of weeks ago on the treatment of severe infection with metallo-beta-lactamases. And again, what they propose is not to treat colonized patients, but to use the combination of ceftazidime avibactam, or the other option is cefiderocol, because in the credible study, some of the patients with carbapenem-resistant Enterobacterialis was due to metallo-beta-lactamases, but we need more data. And the IDSA basically endorsed these recommendations. This is the IDSA document that I would refer you to. What about carbapenem-resistant Pseudomonas aeruginosa? I think this is a very troublesome pathogen that we deal with. You know, Pseudomonas can have intrinsic or extrinsic mechanisms of resistance and inducible mechanisms of resistance. We need to promptly treat the patients because delay in proper therapy increases mortality, and not to forget source control because these pathogens can cause biofilms. And there are many options that are available to us. We have the old options and we have the newer options that are now available, and this was summarized in this recent review. Now, we don't need to be using combination therapy all the time for multidrug resistant Pseudomonas aeruginosa. There are some circumstances where we need to address those which are listed here. We need to consider the co-resistant in Pseudomonas aeruginosa. This is a study that looked at what is the likelihood that when a Pseudomonas is resistant to one beta-lactam for it to be resistant to the other. And as you see, it's much less likely for it to be resistant to ceftolozane tazobactam as compared to tazocin, for example, meropenem or ceftazidime. We know that ceftolozane tazobactam has a very broad spectrum of anti-pseudomonal coverage, regardless of the mechanisms of resistance. And the ceftolozane tazobactam clinical trials done on nosocomial pneumonia, complicated intra-abdominal infections, or complicated UTI, though these were not pathogen-directed trials, but in each one of them, they did have Pseudomonas aeruginosa included, and all three of them showed non-inferiority to the comparator. It's important when we look at the nosocomial pneumonia trial, then when they evaluated the microbiological response and at the test of cure, as you see numerically, those who received ceftolozane tazobactam had a higher microbiological response as opposed to the comparator, which was meropenem. And patients who received ceftolozane tazobactam were less likely to develop emergence of resistance as opposed to meropenem. Real-world data is also showing very favorable results for treatment of Pseudomonas aeruginosa with ceftolozane tazobactam, as you see here, with an overall success rate of 60%. And also here, we need to look at toxicity of other drugs that we have been using in the past for treatment of difficult to treat Pseudomonas aeruginosa. This is data published a couple of years ago, also correlating that patients who received ceftolozane tazobactam were much less likely to develop acute kidney injuries as opposed to those who received polymyxin or aminoglycoside combination. Cefiderocol has also potent in vitro activity against Pseudomonas aeruginosa, and in the credible CR trial, 19% of the patients had Pseudomonas aeruginosa with a mortality of overall for Pseudomonas infection of 18%. These are the recommendations for difficult to treat Pseudomonas aeruginosa by the IDSA, and as you see, they divide them according to the site of infections, and we see that the Infectious Diseases Society of America has listed all the novel beta-lactams. Whereas if we look at the very recently published ESCMID guidelines published 2 days ago, and if we look at their recommendations, basically they listed as ceftolozane tazobactam as the premier therapy for carbapenem-resistant Pseudomonas aeruginosa, saying that there was insufficient evidence available for imipenem, relebactam, cefiderocol, and ceftazidime avibactam. Obviously, time will not allow to go over all these in details. But this is a reference that you can always check. Just a word to say bacteriophages have been used to treat pan-drug-resistant Pseudomonas aeruginosa, but we need more data. They've been only studied in case series, and we need more data to be comfortable using them. Just 2 words about carbapenem-resistant Acinetobacter baumannii. As you know, ampicillin sulbactam is considered appropriate for monotherapy, whereas with all the other agents, it is recommended to use combination therapy. This has been recommended by the Infectious Diseases Society of America to use combination for any other drugs other than ampicillin sulbactam. They give more detailed recommendations about the other drugs such as polymyxin, colistin, as well as the tetracycline derivative, cefiderocol. They're saying that when used cefiderocol, the panel suggests prescribing the agent as part of a combination regimen and not as a single agent because of the results of the credible trial showing higher mortality than the best available therapy. We have sulbactam, durlobactam, which is a new beta-lactamase inhibitor combination, and there are some initial in vitro data as well as preliminary clinical data suggesting that this could be a very good drug to treat Acinetobacter multidrug resistant in the future, and we eagerly await more studies to address this. These are my concluding remarks. I think for the interest of time, I'm just gonna jump to invite everyone to participate in the 32nd International Congress of Antimicrobial Chemotherapy. This is going to happen in Perth, Australia, from the 27th to the 30th of November. Thank you for your attention. And I'm now going to move on to present the next speaker. It's now my pleasure to introduce Dr. Mao Hagihara. Dr. Hagihara is currently an associate professor in the Department of Molecular Epidemiology and Biomedical Sciences at Aichi Medical University in Japan. He completed an infectious diseases research fellowship at the Center for Anti-Infective Research and Development in the USA, and his research interest includes pharmacokinetics and pharmacodynamics of antibiotics. Dr. Hagihara is going to talk to us about basic research regarding novel antibiotics.
Okay. Can you see my slide? Okay.
Yes.
So thank you for kind introduction, Professor Kanj. So I'm very glad to have a presentation today in this webinar. In my presentation, I want to talk about brief introduction. After that, I want to show some data which we conducted with new compound and published in last year. This is a COI regarding my presentation. So since 2019, the SARS-CoV-2, which caused the COVID-19, has been rising around the world. However, from further back, the issue related to antimicrobial resistance has been drawing much attention all over the world, not only from the physician and researcher, but also general people. Similar to COVID-19, the issue related to antimicrobial resistance can impact thousands of millions of people around the world and significantly negatively affecting community health and causing significant economic losses. Antibiotics prevent the millions of deaths every year and remain the primary treatment for the potentially fatal bacterial infections. However, inappropriate prescription rates and overuse of antimicrobials has led to increase the antimicrobial resistance and spreading worldwide, which resulted in the causing global emergency. The number of deaths due to the antimicrobial resistance at least 700,000 in 2013. However, some experts expected that if no action is taken, the number of deaths will be 10 million in 2050. The number is 15 times higher than that of 2013. Additionally, the number is bigger than the number of deaths due to the cancer in 2013. And in these circumstances, major public health organizations such as WHO, CDC in the United States, and ECDC in Europe start to recognize the growing threat of antimicrobial resistance. And they have taken some measure to reduce and prevent these infections. Even in Japan, Japanese government declared National Action Plan on Antimicrobial Resistance in 2016. So we have already known there's some kind of antimicrobial resistant mechanism in gram-negative bacteria, such as purine loss, beta-lactamase producing, and overexpression of Ecrax pump, and so on. The very important thing is some of these antimicrobial resistant mechanisms derive from the plasmid with antibiotic resistant genes. And the plasmid can move to the another bag where they in contact, which called inconjugated, and which result in the making a new antimicrobial resistant bags very fast. Enterobacterales, Pseudomonas aeruginosa, and Acinetobacter species are most frequently detected from the patient as multidrug resistant gram-negative bacteria. Enterobacterales acquires antimicrobial resistant ability mainly due to the beta-lactamase producing and purine loss. For Pseudomonas aeruginosa, purine loss and Ecrax pump overexpression are main cause to be a multidrug resistant pathogens. For Acinetobacter species, similar to Enterobacterales, beta-lactamase producing as the main cause to be a multidrug resistant pathogen. So it is very important to inhibit the beta-lactamases. It's very important to treat of the infection due to the multidrug resistant Enterobacterales and Acinetobacter species. Beta-lactamases can break the beta-lactam rings to deactivate the beta-lactams. And these enzymes, beta-lactamase enzymes, were divided with active site into the serine beta-lactamases and metal beta-lactamases. Additionally, they are divided with molecular class with amino acid sequence of the enzymes into the class A, class C, class D, class B. Which class beta-lactamases the bag detects from the patient is producing is a very important information to choose the appropriate antimicrobials in the clinical situation. The first beta-lactamases was discovered in 1970s. After then, the number of reports have been increasing year after year. In 2018, over 2,700 beta-lactamases had been identified. Some of them cannot be inhibited by established beta-lactamase inhibitor, such as clavulanic acid and tazobactam. Additionally, some of them cannot be inhibited by the carbapenems. The carbapenems was previously regarded as beta-lactamase stable. Carbapenems are mainstay for the treatment of the infection due to the multidrug resistant gram-negative pathogens. However, there is growing concern about the increase of the carbapenem-resistant bags among the multidrug-resistant gram-negative pathogens. Additionally, some previous clinical study revealed that suggested that carbapenem monotherapy may no longer be an appropriate choice for severe gram-negative infections. Carbapenemase has been spreading worldwide, and the prevalent carbapenemase types varied geographically. KPC types dominant in the United States, China, Israel, and part of Southern Europe. OXA48 prevalent in the Middle East, except Israel, and MDM in South Asia. Anyway, the definition of CRE is a little bit different, depending on the region or depend on the country. Especially, at least in Japan, CRE isolate is defined as the isolate showing the carbapenem resistance due to the carbapenemase producing, or AMP-C hyperproduction plus taurine deficiency. This is the result of CRE surveillance, which was conducted in 2017. As a result, 865 cases was reported, and the same number of CRE isolates was collected. And then 28% of CRE isolates have carbapenemase producing genes. Additionally, over 90% of carbapenemase producing genes are classified into the IMP type carbapenemases, mainly IMP1 and IMP6. This is a list of new antimicrobials recently approved by FDA. A red character shows the antimicrobial targets for the gram-negative bacterial infections. From 2014, 4 new beta-lactam and beta-lactamase inhibitor combined antimicrobial was approved by FDA. Then, among them, ceftazidime and avibactam, meropenem and bavobactam, and impenem and relebactam are allowed to use for the CRE infections. However, even now, the CRE infection shows a relatively higher mortality, especially for bacterial infections. Diazobicyclooctane is a new class of beta-lactamase inhibitor, which shows has strong inhibitory activity against the beta, especially for class A and class C beta-lactamases. As I mentioned, avibactam and relebactam are currently used in the clinical station, while depending on the country. On the other hand, nacibactam and zidebactam are under developing, especially for nacibactam, which compound we use in our in vitro study. I will show some data. This chemical structure attached here. As a result, this compound has antimicrobial activity by itself. This is the list of inhibitor activity of various beta-lactamase inhibitors against the beta-lactamase enzymes. Nacibactam showed inhibitory activity against KPC, which point is different with tazobactam. Compared with avibactam, bavobactam, and relebactam, nacibactam showed that very similar inhibitory activity against the beta-lactamase enzymes, especially for class A and class C beta-lactamases. Nacibactam shows strong inhibitory activity. This is a result of the in vitro study to evaluate the affinity to the penicillin binding proteins. Compared with the other beta-lactamase inhibitor and beta-lactams, nacibactam show relatively lower IC50s, which means that nacibactam has a high affinity to the penicillin binding protein 2. This is the main reason why we are thinking that this is the main reason why nacibactam has antimicrobial activity by itself. This is an in vitro study data. In the study, researchers explored some kind of concentration of diazobicyclooctane, which shows the x-axis. At the same time, researchers exposed various beta-lactam agent to the E. coli with beta-lactam as a negative and Nacibactam resistant mutant. And then they evaluated MIC, especially when piperacin and Severin and aztreonam was combined with Nacibactam. With higher concentration of Nacibactam, the researcher admitted a lower MIC of beta-lactams. This result suggested that Nacibactam works as an enhancer of the antimicrobial activity of various beta-lactams. This is an epidemiological study of the CRE, which was conducted in Asia. E. coli and Klebsiella species and Enterobacter species were frequently detected as a CRE isolate from the patient. Then we focus on the respiratory specimens. Enterobacter cloaca shows the secondary highest detection rate among the CRE isolate, followed with the Klebsiella pneumonia. This is our research flow, research flow. At first, we conduct an MIC study with aztreonam, Severin, meropenem, and Nacibactam against the 14 isolates, including Klebsiella pneumonia, E. coli, Enterobacter cloaca. In parallel, we conduct a PK study. Then we evaluate the PK profile in plasma and ERF when antimicrobials was dosed as monotherapy or combination therapy of beta-lactam and Nacibactam. Using these data, we conducted a PD study with site infection model and pneumonia model. Then we evaluated bacterial density change after 24 hours from the start of antimicrobial therapy. Then finally, we conduct a PK-PD study using these old data. Then we evaluate the relationship between antimicrobial activities of several regimens and the PK-PD parameters. This is a result of MIC study. In our study, we included some kind of CRE isolate. They have some various beta-lactamase producing genes, such as KPC2, IMP1, IMP6, CTMX, and OXO48. As a result, we admit a combination regimen show 128-fold to 2-fold lower MICs when compared with beta-lactam single regimen. Even against CRA isolate with class B beta-lactamase producing genes, combination therapy shows a lower MIC, significantly lower MICs against the CRA isolate. We are thinking that the reason why Nacibactam has an antimicrobial activity by itself due to the high affinity to penicillin binding protein 2, while Nacibactam cannot inhibit the class B beta-lactamases. This is the result of the PK data of Nacibactam, meropenem, and Severin and aztreonam in ERF with black line, in plasma with dot line. To use a human simulated doses of the 1 gram Q8 of beta-lactams and 0.5, 1, 2 gram Q8 for Nacibactam in our in vivo study. Based on this data, we calculated and we use adjusted dosage in our in vivo study. This is the one result of PD data in site infection model. Y-axis shows the result bacterial density change after 24 hours. White bar shows the result of control. Blue bar shows the result of the beta-lactam monotherapy. Light gray, the gray, the black bar shows the result of combination therapies. As you can see, combination therapy shows a significantly lower bacterial density after 24 hours, which means that combination therapy shows a significantly higher antimicrobial activities. against CRE positive Klebsiella pneumonia. So while I didn't show the data, when we use CRE positive E. coli, we admit a similar result. And this is the one result of PD data in lung infection model. As we have expected, combination therapy shows significantly higher antimicrobial activities compared with beta-lactam monotherapies against the 4CRE positive Klebsiella pneumonia. Additionally, we admit a similar trend, similar result when we use a CRE positive Enterobacter fluorata. This is the result of PK/PD analysis in side infection model. Finally, we found time above MIC of beta-lactams correlated the antimicrobial activity for each regimen very well, compared with Cmax per MIC and AUC per MIC. With higher time above MIC, we admit a lower bacterial density, which means a higher antimicrobial activities for each regimen. And to attain the one log reduction of bacterial density at the infection site, at least 20 to 30% of time of MIC are needed for each regimen. This is the result of PK/PD analysis in lung infection model. As you can see, very similar result. We admit a very similar result with the result in side infection model. Time above MIC are correlated with the antimicrobial activities for each regimen very well. And with higher time above MICs, we admit a higher antimicrobial activities. Even when we use our ELF data, ELF PK data, and the plasma PK data was used as PK data. This is a summary. CRE and CP have become a global threat now. And this infection has been associated with high mortality due to the limited treatment options still now. However, our study reveals the combination regimen with beta-lactam and nacibactam are preferable candidates to treat infection due to CRE and CP. Additionally, this fact can give new therapeutic choice to antibiotic treatment for CRE infection, while the prevalence and burden of CRE infection are rising. Thank you very much.
Thank you very much, Dr. Hagihara. And last but not least, we will hear from Dr. Hiroki Kitagawa. In 10 minutes, he will present to us on the topic of ultraviolet disinfection for infection control. Dr. Kitagawa is currently an assistant professor in the Department of Infectious Diseases at Hiroshima University Hospital and has a Doctor of Philosophy in Medical Sciences. His research interests include the control of multidrug resistant organisms and the use of the UV light. Dr. Kitagawa, the microphone is yours.
Thank you for your kind introduction. Can you see my slide?
Yes, we can. Go ahead.
Okay. I'm Hiroki Kitagawa from Hiroshima University Hospital. Today I talk about ultraviolet light disinfection for infection control based on our experience. Healthcare-associated pathogens can persist on inanimate surfaces for weeks or even months. Inadequate manual cleaning can be directly linked to an increased risk of hospital-acquired infections. Common route of transmission of healthcare-associated pathogen. The main route is hands of healthcare personnel, patient to other patients. However, environmental healthcare environment, including isolation room surfaces, portable equipment, are major important source or transmission of healthcare associated pathogens. So recently, non-touch disinfection systems have been increasingly introduced as an additional approach to manual cleaning. Non-touch disinfection system mainly included ultraviolet light and hydrogen peroxide vapor. Now, there are several types of devices which we can use. In our institution, we have introduced pulse xenon UV disinfection devices. We performed a PX UV disinfection after manual cleaning in all patient rooms after discharge or transfer in ICU, ECU, and HCU. PX UV disinfection was also performed in isolation room, in other words. This device emits broad spectrum UVC light at the pulse frequency, like these pictures. To reduce shadow areas, the patient room were disinfected using 2 5-minute cycles of PX UV per room, with 1 cycle performed on each side of the patient's bed, this side and this side. In the patient rooms with separate bathrooms, 1 additional 5-minute cycle was performed in the bathrooms. We previously investigated the in vitro effect of the PX UV disinfection. After applying the bacteria on the plate, the right side of the plate was covered, and after that, plate was irradiated with UV light. And in this picture, left side was irradiated with UV. MRSA was completely disinfected. However, right side, the shadow areas are ineffective for UV disinfection. This is a very important point for UV disinfection. Shadow areas are not ineffective. This picture shows the effect of UV disinfection for CPE. UV disinfection is similarly effective against CPE. However, C. difficile spore were more resistant to UV disinfection, so a longer irradiation time is required for effective disinfection. In Japan, MDR negative bacilli, including CP, is not so frequently isolated. However, a history of treatment or hospitalization abroad is a risk factor associated with the colonization or infection of MDR bacteria. And nosocomial outbreak originating from these patients have also been reported. We experienced a patient with an infection caused by NDM positive Klebsiella pneumonia with a history of hospitalization abroad. After the discharge of this patient, environmental contamination of this organism was observed on the surface of this patient's room, even after terminal cleaning, especially around his bed and around the sink. After PX UV disinfection, this organism was not detected. In Japan, MRSA is the most common isolated MDR organism. So we investigate the MRSA contamination in the patient rooms. Private rooms occupied by the patient infected or colonized with MRSA. High-touch surfaces were sampled using contact plates before manual cleaning, after manual cleaning, and after PX UV disinfection. This figure shows a proportion of MRSA positive samples. About 20% of samples were positive for MRSA after manual cleaning. PX UV disinfection significantly reduced the proportion of MRSA positive samples compared with manual cleaning alone. We also investigated the instance of MRSA acquisition before and after the introduction of PX UV disinfection. PX-UV disinfection was performed after manual cleaning in all patient rooms after discharge or transfer in ICU, ECU, and HCU of our hospital. Outcome was the instance of new infection or colonization of MRSA. The incidence rate of MRSA acquisition have been decreased in intervention units. However, non-intervention units, the rate was not changed. Statistical analysis shows the PX-UV disinfection significantly decreased the risk of MRSA acquisition. Conclusion, UV disinfection can reduce the environmental contamination of MDR bacteria. Strategies for effective use of this technology to reduce hospital-acquired infection need to be investigated. Thank you for your attention.
Thank you, Dr. Kitagawa. And now I think I'd like to go over some of the questions that were submitted to us. I see that Professor Paterson had to leave us because he had another commitment, but he already answered a couple of the questions on the chat. So I'm just gonna read them for everyone to be aware of how he answered. So one of the questions was resistance to common antibiotics such as ceftriaxone is becoming more frequent. What treatment options would you advise? And then resistance, this poor hydrolytic profile obscures the detection of OXA48 like. It may go undetected in routine diagnosis and complicate the treatment option. What are your thoughts? And basically what Professor Paterson answered is opinions vary. I would avoid a carbapenem even if the lab tells us that it is susceptible. I think Dr. Severin had also a comment on this question. Would you like to answer it verbally, Juliette?
Yes, thank you for this question, by the way. It's a good question regarding the detection there. I put a nice review paper in the chat box, and there are many suggestions that can be followed. We use the ertapenem MIC as a screening, if it's elevated MIC as a screening to continue further testing. Regarding the treatment, I think that's a very difficult question, and even in my country, we have debates about it. So other opinions, I'm glad to hear because I think it's a difficult topic. Yeah.
Yeah, absolutely. I mean, for OXA48 right now, I mean, if in countries where they have ceftazidime, avibactam, there are some real world data suggesting that it does work, but we need to await more studies to confirm that. Cefiderocol might have activity as well, but again, we really need to look at more studies to confirm that. I must add that in some countries, such as my own country, Lebanon, Turkey, many countries in the Mediterranean area, OXA48 is the predominant carbapenem-resistant Enterobacterialis, much more than KPCs, and we do have some NDMs, but OXA48 seems to be the predominant. And so we're gaining more experience with using the novel beta-lactamase inhibitors for such bacteria. I think there are a couple of questions for Professor Yanagihara. So one of them is with the Verigene molecular diagnostic assay, do you encounter any test failures such as inability to obtain the test result because of high variability in the target specific signals?
Very good question. because it's very important. So we have to understand that molecular diagnosis and the culture-based current diagnosis is different. So it's also that we saw that the difference between two test results. So you have to think about it. So we see that we encountered that case. So it depends on the situation. But in the future, so we receive that two information. One of that molecular diagnosis, the other one is a culture-based diagnosis. So we can compare and we can think about it. That's very good question.
Thank you very much. Another question related to what do you do when you have a polymicrobial infection, and how do we go around this by using the molecular techniques?
Yeah, also this is a very good question. So also we have to think about that it's different between the molecular and culture-based. If we use culture, so we can take all bacteria, including the normal flora. But molecular diagnosis pick up some of them. And I will answer his or her question. Polymicrobial, the detection level is 10 to the 5th power, I guess. So the Staph aureus, this is at the 10 to the 5th power, and enterococci, 10 to the 5th power. We can get two data. But the other one bacteria, for example, the Pseudomonas aeruginosa, 10 to the 7th power, and the other one, E. coli, 10 to the 4th power. So it cannot detect the 10 to the 4th power E. coli. It's happening. So also we can think about the situation.
Yeah, and a last question for you before I have a couple of questions for the other speakers is there was a publication a couple of days ago on using the gram stain as a stewardship practice to decide on antibiotic therapy and pneumonia. So they're asking, do we still need the molecular diagnostics, or can we just depend on the gram stain?
That's also very good question. I understand gram staining is important for that VAP because we can take the sample such as that bronchoalveolar lavage and sputum. We can see that the gram staining mentions at the gram positive or gram negative. So if you see that the gram positive, you use that vancomycin for MRSA. But if you don't see that gram positive, so you don't have to use vancomycin. But if gram positive, maybe not MRSA, maybe MRSA. But if the Staph aureus is not MRSA, it's MSSA, we cannot use that vancomycin. Or same thing happening at the gram negative. Right. It's my question, my answer.
So maybe they complement each other, but obviously they have significant added value. Question to Dr. Kitagawa. They're asking about how does the hydrogen peroxide vaporization compares to the UV light? Do you have any experience with that?
Thank you for good questions. The hydrogen peroxide system is also effective for the MDR bacteria. However, the characteristic is different from the UV disinfection device. The hydrogen peroxide vapor system need a long time, about 1 to 2 hours per rooms, and we can use this device open space, so the closed the private room only. In contrast, UV disinfection system can use open space with some curtains, some other equipment, and it takes a short time, about 10 to 20 minutes per room. So the characteristic is different. So we know the characteristics of each devices. So,
okay, thank you very much. And a question to Dr. Hagihara about when do you think Nacubactam combinations will enter into clinical trials? Do you have an idea about whether we should expect randomized clinical trials soon with this compound?
I'm sorry, I don't have any information. It is a very secret for the drug company, right?
All right.
So I have no information now.
We don't. And the last question to stay on time is for Professor Severin. Should we be using the difficult to treat definition for Pseudomonas aeruginosa? It's been used more recently, DTR when it's resistant to all except all beta-lactams, including carbapenems. Do you think we should be using DTR definition or not so much?
That's a good question. I just recently discovered that this definition was actually used. The way I read it, but maybe there are different definitions around, was that it's a combination of the beta-lactams plus the quinolones. I don't know. It's just another definition in the field, right, and it keeps on confusing things. I think it's one of my last sentences in my presentation was that maybe we should just focus on bug-drug combinations. Right. And what you see is also that a lot of presentations today are around these beta-lactam and certain bacteria, especially for treatments, etc., but also for IPC measures. So I think these drug-bug combinations are more useful than this difficult to treat definition, but yeah.
Yeah. All right, well, to stay on time, I'd like to really thank all the speakers for their excellent presentations. I'd like to thank all the attendees who participated, attended, and sent us their very interesting questions. Again, it's been my pleasure and honor to chair this webinar on behalf of ISAC and the Japanese Society for Chemotherapy. Thank you very much for all of you for attending, and I hope to see you in the future. Have a good day. Bye-bye.